Okay, everyone, welcome. So today we're going to be covering DNA and gene expression. And so taking a relatively deep dive into these two topics. So let's get into it. So as we all sort of know, cells have our DNA or deoxyribonucleic acid gives the macroscopic level, so large scale, as well as the microscopic level, and everything in between. And so those set of structures is the DNA. The DNA, in at least in eukaryotic cells, is found in the nucleus, and our DNA is organized into what we call chromosomes, which are essentially short um, segments of DNA, oftentimes circular or linear, depending on the organism. And then the final thing I'll make mention of is that all of our sort of information, how we take that DNA and put it into action, as you will recall, is simply referred to as a gene. Now, we have sort of this idea that every cell, at least in a eukaryotic cell, and specifically we'll think mostly about humans here, that our DNA is comprised of distinct molecules, and our molecules undergo distinct periods of replication, allowing us to generate overall 2 copies for each daughter cell, allowing us to pass DNA not just from generation to generation, but also from 1 cell to 2 cells and 4 cells and so on and so forth. So let's just look at sort of a schematic here, right? We have the nucleus, the cytoplasm. So this is a DNA, I'm sorry, a eukaryotic cell, obviously because it's membrane bound organelles. And then we have DNA, which we can replicate it, becoming 2 parts. And we'll talk about mitosis, i.e., the replication of DNA, in a subsequent class. But the other aspect of this is what we'll call the central dogma of molecular biology, where we have transcription of DNA into messenger RNA, and then translation of that messenger RNA into protein, thus the action. And so these are our 2 overall processes we're really going to be focusing on today, transcription and translation. Um, and so before we sort of move on, just a few slides for you. They're optional slides. I recommend taking a peek, a little bit about the history of DNA, how it was discovered, what are some of the major milestones, some controversies, and things like that, as well as some pretty cool experiments with some bacteria that I definitely recommend you take a peek at. Because again, history is obviously a really important part of the discovery of DNA, but also just knowing about science, knowing how it's discovered allows you to appreciate it and understand it more. So again, definitely take a peek at those 20 or so slides about the history of DNA. It's really fascinating look in history. Lots of interesting characters. And if you're a lady in science, lots of fantastic female scientists along the way as well. So DNA has an overall basic structure to them. Now, we found out that DNA, and very early on, that DNA is comprised of 4 DNA bases: adenine, thymine, guanine, and cytosine, or AT, G, and C. Now, we know from DNA that there is complementary base pairing. This is figured out by Erwin Chargaff. And Erwin Chargaff has his rules, simply just called Chargaff's rules, where A will only bind to T and G will always bind to C. Thus, when we have complementary base pairing. And so we can sort of look at sort of some ratios of A's and T's and G's and Z's. Now, you ask yourself, why is there some variation about it? Well, remember, DNA is not perfect, right? It's not always copied perfectly. It's not always 100% accurate. So there is some variability in that one-to-one ratio. So we have Chargaff's rules, and sort of the interesting thing about Chargaff's rules is he kind of sort of came up with them, but he didn't quite realize the importance of it, and not until Watson and Crick, as well as some other scientists, really heavily proposed the idea of the double helix model, which we'll get into in a second. Now, you guys have all probably heard of Watson and Crick, 2 really influential scientists. They essentially discovered the structure of DNA, and they discovered a couple key things about it. Again, building off the work of others. First was that DNA has a sugar phosphate backbone, and each of those backbone pieces has a nitrogenous base attached to it. And they proposed that the DNA was not just a straight piece of linear information here, that it was in fact that double helix. Now, they got that double helix actually from a really awesome female scientist called Rosalind Franklin. Now, Rosalind Franklin oftentimes overlooked person in sort of the field of the discovery of DNA, but her really influential X-ray crystallography pictures of the structure of DNA, as you can see here in the top figure, mimic exactly what we know to be the actual 3D structure of DNA. So DNA, remember, it's a double helix. It's a ladder, but a ladder that is twisted in and upon itself. Now, there's a sort of overall structure here that we'll look at. And again, this should all look pretty familiar. Now, DNA has polarity. Now, when you hear the term polarity, it means essentially the same thing as when we were talking about microtubules, or the same thing when we were talking about protein sequences, right? It simply means polarity means there's directionality to it, right? Whereas protein has N terminus and C terminus, and our microtubules had a plus end and a minus end. Our DNA has a 5' end and a 3' end. Now, what the 5' end derives from, as you recall, is the 5' end has the 5' carbon, so carbon 1, 2, 3, 4, 5, thus the 5' carbon, and then the 3' end, carbon 1, 2, and 3. Thus, this is why it's called the 3' end. This is called the 5' end. If you sort of need a refresher on what that's like, this is what we covered in chemistry 2, just as a note. Now, some overall other structures about DNA besides its polarity. Again, we have that complementary base pairing here. So we have that adenine with thymine, cytosine with guanine. Sorry about that. And they are nitrogenous bases. They are very, very rich in nitrogen. So if you ever wonder why do I need nitrogen in my diet, it's mostly to make DNA. But again, the other important aspect here about DNA is it's got a 5-carbon sugar, as well as a phosphate, thus making the backbone. Now, the backbone is joined together by what we call phosphodiester bonds, joining, as you recall, this is a phosphate or phospho. And then we have ester, POC. Anytime you have an oxygen in between two distinct things, it's called an ester. And so this is what we have called a phosphodiester bond. That's the overall structure here. And then the final thing is the size, right? We're looking at 2 nanometers for the width of the double helix. And we have between a major groove, which is we define as a large gap here, and a minor groove, which is a smaller gap here. If we encompass one major groove and one minor groove, essentially one full 360-degree spin of our DNA, we get a length of 3.4 nanometers. So lots of things, important things going on here with DNA, but it's also a very small structure, but clearly a very important structure. Now, the final thing I'll make mention of, actually two final things I'll make mention of, is the first is these blue dotted lines. You recall dotted lines in between two atoms implies that we have a hydrogen bond. So HO and H, those are 2 hydrogen bonds, and hydrogen bonds seem to be the most important factor here in holding our DNA together. So strong, but not impossible to break, and that'll become you throw your ball at your friend throws a ball back. They're parallel to one another, but they're moving in opposite directions. And we see the same thing for DNA. So that's our overall structure of DNA. Clearly, we spent a lot of time on it because it's important. You got to remember the structure of DNA. It is. I can't understand how important it is to know DNA. So let's talk a little bit more about DNA size. Now, these sort of what were in these past few slides that I sort of skipped over. They basically talked about what I exactly said. Now, DNA is measured in what we call base pairs, or BP. Now, we can also scale that up to kilobases or kilobases, depending on how you say the word. Doesn't really matter. Where we have thousands of bases. So 1 kilobase is equal to 1,000 base pairs. So when we say a base pair, what we're referring to is just one of these bad boys, right? 1 thymine, 1 adenine, and all the backbone attached to it. That is what 1 base pair means. Now, 1,000 base pairs, or a kilobase, is just 1,000 of those units. That's it. So oftentimes in lab, for instance, we'll see, oh, this PCR amplifies a 500 base pair section of DNA. Well, that simply refers to a piece of DNA that is 500 bases long. That's pretty much it. Now, DNA has some slight variations to it. Now, we have what's called right-handed DNA, and this is what we call bDNA. Now, bDNA are naturally occurring. We see them in the environment. This is by far the most common type of DNA that you see, that you kind of know and love. And then we have this funky one, which is left-handed DNA. which, you think, oh, chemistry has always got to be fun, right? And of course it does, right? But zDNA is left-handed DNA. So let's take a peek at bDNA. So bDNA, as you'll notice, spins like so, right? That overall shape here. And then we have contrast to that, we have zDNA, which has a completely different shape, right? It's just very, very different. Now, there's a couple other ones I'll just make mention of. We have aDNA. It's also right-handed, but it's shorter and thicker than bDNA. And so that's just kind of another funky thing, just as a note. All right, so funky structures of DNA. What we care about most is clearly bDNA, right? Most students generally don't see zDNA until they get to until they get to cell biology with me. So, not, if this is what you need to sort of think about, but do be aware of zDNA as well as aDNA. Now, DNA, it's very, very long, right? It can be thousands upon millions and billions of base pairs long, and cells are small, right? The nucleus of your average eukaryote is about 20 microns in that ballpark, so not a very large area. I'm sorry, a very large area or large volume. So DNA has to be oftentimes supercoiled, and supercoiling simply refers to wrapping something really, really tight around something. Now, DNA can be twisted further in what we call the same direction, so just spinning, say, all clockwise, and we call this a positive supercoil, or we can twist the DNA in the opposite direction that is already coiled, and this is what we call a negative supercoil. Not super duper important, but just to sort of make mention of those two terms. Now, DNA, sort of what we have here, where we have a relaxed piece of DNA, and we have a positive supercoil and a negative supercoil here, sort of cartoon schematics, but we can see the overall form here, right? Negative supercoiling here, and then relaxed DNA. And clearly, the picture on the right takes up far more space than the picture on the left. So the idea being that DNA, when we supercoil it, can essentially reduce overall volume, reduce overall area, allowing us to pack a lot of DNA into a small area. Now, there's an enzyme out there, and we know it's an enzyme because it ends in ASE. These are called Toposoma Races. Now, Toposoma Races are cool because they can either relax or tighten supercoils, so they do both. Now, there's two distinct classes, type 1 Toposoma Races, which induce what we call a transient single-stranded breaks in DNA, so they just breaks, right? And then we have type 2 toposomerases, which actually introduce double-stranded breaks. And an example of this one is actually DNA gyrase, which we'll come back to very shortly. So this is just a schematic of how these toposomerases work. So type 1s cause a single break, allowing DNA to rotate and pass through the break, then resealing it. Or we have a type 2, which causes essentially a double-stranded break and allow the cell to do whatever it needs to do with the DNA. So the other thing to make mention of is that DNA can be broke apart. It can be very much like proteins, right? Proteins have a structure, and when you denature a protein, you change its structure. So we can. No, I just don't want you. So what we can see here is that DNA can break apart very much like proteins. Now, DNA can easily be separated because conceptually, right, DNA is mostly held together by hydrogen bonds. And as we know, increasing temperature, changing pH, and many other factors can cause hydrogen bonds to easily break, right? That's the purpose of hydrogen bonds. They're strong, but not super duper strong, like say a covalent bond. So we can denature our DNA. And we can actually look at this and calculate DNA melting temperature, or what we call T sub M. And it actually varies depending on the content of of A's and T's or G's and C's. So we can look at the absorbance of DNA. Now, it's interesting thing about DNA, it does absorb light with a maximum around 260 nanometers of the wavelength of light. And a sort of interesting thing about this is as those strands separate, so DNA breaks apart, the absorbance increases rapidly. And so the idea here is we can look at relative absorbance over temperature. And we can see that at 70 degrees, the relative absorbance is 1, but by the time we're at boiling point, as we go from double-stranded to single-stranded DNA, the temperature increases, thus the absorbance increases, as I made mention of, and our TM is actually about essentially the temperature at which half or so of the DNA has been denatured. And so you might ask, why do we care about TM? Well, I do bring it up because we do cover it in lab, right? When we do polymerase chain reaction in lab, we actually use the TM of our DNA to actually make PCR work. So just as an FYI, which is why we're covering this. And the cool thing here is actually you can look at TM over a temperature gradient. So we have 60 to 100 and a bunch of different organisms. So we have a couple bacteria, we have calf, salmon, bacteriophages. I forget what amphibia is, but it doesn't really matter so much. But you notice there is a linear correlation between TM and the amount of guanine in cytosine. Now you might ask yourself, why does that work? Well, let me go back a couple slides and we can take a peek at that of why the more guanine and cytosine you have, the stronger your DNA is. Well, look how many hydrogen bonds are in guanine and cytosine versus how many bonds are adenine and thymine. So if you got to do your best guess should probably tell you that 3 hydrogen bonds is stronger than 2 hydrogen bonds. So when you have a here, it's a group of bacteria known for causing some pretty interesting infections, likes hot temperatures. It likes temperatures over 40, and it likes temperatures over 40 for a lot of reasons, but the reason it can stand temperatures over 40 because it has a high TM of its DNA, which is again tied directly to its GC content. So let's talk about how DNA is packaged. Now, packaging DNA is tough, right? It's not just tough for us where we have billions of base pairs of DNA, but it's tough for bacteria as well. Now, bacteria, as we know, they don't have a nucleus, they don't have membrane-bound organelles, right? They're prokaryotic. However, they still have to package their DNA, right? They're only so big, and because they're only so big, they still got to package that DNA. And it's sort of the reality here is like if you stretched out your DNA in your cell, for instance, it would be meters long. That's how much DNA you have in a single cell. It's kind of an amazing thing. So in our bacteria, all of their DNA is much smaller than ours, but it is packaged in a single circular chromosome. Now, there are some funky bacteria out there that have multiple chromosomes, and there are some bacteria that have linear chromosomes. But your average everyday bacteria, as well as archaea for that matter, have a single circular chromosome. Again, there is a lot of variability there, but again, typically speaking, a single circular chromosome. Now, the DNA in bacteria is bound to small amounts of protein, and it's going to be similar to histones, which I'm sure you probably recall from Bio 1. But there are these proteins that the bacteria wrap their DNA around to supercoil it, make it more condensed, leading us to what we call a nucleoid or the nucleoid region of the cell. So we can look at a bacteria here. You can see its nucleoid is here. You can see its DNA tightly wrapped around itself here, not taking up all of the volume of the cell, which it theoretically could, but it doesn't because it's super coiled around a group of proteins. Now, conversely, you could take that bacteria and you can split it open, right? Lyse it, and it could spill all of its DNA out. And again, you can see how much DNA a single bacteria has. It's pretty remarkable, right, when you think about it. Now, how does it get that? Well, it packs everything around protein in what we call the nucleoid region. Now, bacteria also have an interesting piece of genetic information called plasmids. Now, plasmids could be their entire class or their own class, just as a note, because they are wild and wacky things. But they do carry lots of interesting things like heavy metal resistance genes, antibiotic producing genes, antibiotic resistance genes. There's lots of things that these bad boys cover. I'm sorry, contain, not cover. Now, bacteria have a couple different types of plasmids. So they're F or fertility plasmids, and this is actually involved in the process of conjugation, which we simply term as bacterial sex. We have resistance plasmids or R plasmids, and these actually plasmids that carry genes that give them resistance to drugs or metals or things like that. So they are beneficial for the ecology of these organisms or pathogenesis if they're pathogenic organisms. And then we actually have call or collagenic factors, which actually allow the bacteria to produce collicins, which actually kill nearby bacteria that lack call factors. So we're not really going to talk too much about, we're not going to talk about those any further, I should say, but I do want to make known that there are different types of plasmids. So I'm sure you probably learn a little bit more about those when you take microbiology with Professor H. So bacteria are simple, but we are complex, so we're much harder. Now, when our eukaryotic DNA, when it's bound to proteins, these proteins are called histones, the DNA is converted into what we call chromatin. However, at the time of cellular division, our chromatin fibers will condense into a much more compact structure, which we simply define as the chromosomes. And so let's, oh, I got rid of the picture, never mind. So let's talk a little bit more about the proteins that our DNA wraps around. These are called histones. Now, histones are interesting because they're pretty small. They're only a few hundred amino acids long, but they are very, very abundant in a eukaryotic cell, and they are rich in lysine as well as arginine. Now, what you'll recall is that our friends lysine and arginine are pretty commonly as amino acids. They are charged, right? They are in the group of charged amino acids. Now, DNA always carries a negative charge on its backbone. So, because of the negative charge of DNA and the positive charge of the proteins, our histone proteins, DNA strongly binds to those histones. It's kind of cool. Now, the final thing I make mention of, and I always think this statistic is wild because the mass of histones in a single eukaryotic chromosomes is actually equal to the mass of the DNA. So when you look at your DNA, it's about equal amounts protein as it is DNA, which I kind of always think that's kind of cool. So there are 5 main classes of histone proteins: H1, H2A, H2B, H3, and H4. Do I expect you to remember that? Absolutely not. Just to let you know. But just to know that that is what they're called. Now, chromatin, which again is a protein DNA hybrid, right? Chromatin has about equal numbers of all of those except for H1. Now, H1 is relatively rare. Well, I should say not relatively rare. It's less common than the other 4, H2A and H2B and so on and so forth. So that's just as a FYI. You don't need to know that, but it's just there because it's true. So let's take a peek at what we have here. We have a what's called a nucleosome, which is going to be a set of histone proteins with DNA wrapped around them. And you see all the little bits and pieces here in the middle. That's DNA. Now this looks, and many people have described it as such as like beads, right? If you have like beads on a string or like a necklace or a bracelet with beads on it, that's exactly what DNA really looks like. Now the final thing I'll make mention of this figure is the size, 0.05 microns or micrometers, depending on how you want to say it. So we're talking about 50 nanometers size here, right? So not very big, not very big at all. So very, very small, but obviously has to be small if we're going to package our DNA. So let's take a peek at our friend here. So we have our nucleosome. There's 8 histone proteins, so 4 on the top, 4 on the bottom. I'm sorry. Uh, 8 histone proteins. Sorry, 4 on the top, 3 on the bottom, and then we have H1 here. So you can see we have 2 H2Bs, 2 H4s, 2 H3s. There's an H2A. There's only one H2A, I should say. And DNA is wrapped around it not once, but twice. And you notice H1 does associate with the side here. Then we have what's called a linker piece of DNA, which connects our 2 nucleosomes together. And then overall, again, we're looking at a size of each nucleosome being about 10 nanometers. So nucleosome formation is just the first part, right? So we start to wrap them around these histone proteins, and these chromatids, which again look like beads on a strings, actually measures, as I mentioned, about 10 nanometers in diameter. But we can actually have the chromatin of intact cells measures at about 30 nanometers, or what we call the 30 nanometer fiber. So let's take a peek at sort of the overall structure that we've walked through here. We start with the double helix. We wrap around our histone proteins at 10 nanometers, and then we form what's called the 30 nanometer fiber, which is again held together by H1. So you sort of see H1 here. It's kind of off to the side. H1 helps sort of wrangle a bunch of histones together to form what we call that 30 nanometer fiber. Again, just not just having these beads, but it's essentially taking these beads on a string and essentially pushing them on top of one another, creating a much denser structure, 3 histones tall. So you notice we go from 10 nanometers to 30 nanometers. Then from there, we'd start to see what's called DNA loops, where we create these overall larger loops here. Allowing, you can sort of see the little size piece, right, as it goes up and up and up, where one of these chromatin fibers makes up one segment of our overall loops. And again, we're getting much larger, so 300 nanometers. And then from there, we start to go into what's called heterochromatin, where we start to supercoil and wrap things tighter and tighter and tighter. And then we finally get to the highly condensed chromosome structure that you know and love, where there's a centromere, 2 distinct chromatids. And again, it all builds up by wrapping first around nucleotides. And so there are some important aspects to how we package DNA. Now, you can imagine like packaging DNA like this, or packaging DNA even tighter than that, right, might make it kind of inaccessible. right? You can imagine like if you took a, for instance, like a, I don't know, you had a, say a toy same way. When DNA is open as a double helix or even as a nucleosome beads here, it's easy to get to. When we start to get to these higher levels of structure of DNA, it becomes a little bit more difficult to get at. And because DNA is important for transcription, the tighter DNA is packed together, essentially means the harder it is to get. And the harder it is to get at DNA, the more inactive it will be. So the tighter DNA is wrapped around histones, the more inaccessible it is. And the looser it is, the more accessible it is. So there's a relationship between how tight it is and how accessible it is. The tighter it is, the less accessible. The looser it is, the more accessible. Uh, which is kind of a cool thing. Now, organisms, and it doesn't really matter who it is in the world of eukaryotes, they can alter their chromatin packaging, right? They can all essentially alter how tightly it is wrapped around. Now they can do this through a couple different mechanisms. Now, the first is what we call histone methylation, and the second one is histone acetylation. Now, histone methylation is when we tag a methyl group, so just a CH3, on a lysine via an enzyme which is called histone methyl transferase. So to break that down, histone, i.e. the proteins, methyl as in a methyl group, so CH3, transfer as in I transfer from one to the other, and then ACE as an enzyme. So it's an enzyme that catalyzes the transfer of a methyl group to a histone. It's pretty straightforward. Now, what methylation does is actually serves as a signal for activation. So when you have lots of methyl groups on your DNA, it can signal either for the activation or the repression of transcription, depending on the lysine that's involved. Now, we can also acetylate histones via a protein called an acetyl transferase. So it transfers an acetyl group, uh, an enzyme that transfers an acetyl group. Um, Apologize for the dog in the back. Okay, okay. Sorry about that. Someone had to deliver a package, and when people deliver packages at my house, it is time for an all-out bark fest. Okay, so methyl transferases transfer methyl groups. Acetyl transferases transfer acetyl groups, and there's also a final one which is called a histone deacetylase, which removes acetyl groups. Now, what this does is it actually modifies our chromatin. We call this chromatin remodeling, and it can either open it, so i.e. wrap it less tight, or close it, wrap it tighter, calling it either active or inactive. So it's open, it's active, closed, inactive. Now, there are some other proteins I'll make mention of, which we call chromatin remodeling proteins, which actually alter the position of nucleosomes. For instance, there's a really important class of remodelers called SWI/SNF family, which actually can slide or completely remove nucleosomes from a region of chromatin, making DNA more successful or accessible. So those are some enzymes involved. That's some overall ways to loosen or tighten. But the question is, why do we care? Right? Well, remember, the tighter it is, the less accessible it is. And we care about DNA not because it's fun or anything, right? We care about it because it's the blueprint for life, right? We need it to transcribe. And so sections of chromatin that are really tightly compacted, and they actually show up as dark spots in electron micrographs, which is actually kind of cool, are called heterochromatin. So thus they're inaccessible. More loosely packed DNA, which has and is very, very diffuse, we'll call that euchromatin. Chromatin becomes highly compressed into heterochromatin. But when we're talking about cellular division, just as a note, right? It might condense into heterochromatin, but it will come back to euchromatin after the fact. Now we have a couple other sort of terms that I want to make mention of before we move on. The first is facultated heterochromatin, which can actually be converted to euchromatin and vice versa. So it kind of can hop back and forth between active and inactive. We also have constitutive heterochromatin, where actually some heterochromatin is permanently compacted, making it completely inaccessible, which is kind of a cool thing. Now, two of these constitutive heterochromatin that you've heard of, but maybe have not heard of them be called as constitutive chromatin, is actually what we call centromeres and the telomeres. Now, your centro is center, telo is end, so center and end of chromosomes. So we can look at this, our centromeres here, and then we have a telomeres here. Two areas that you can see under these staining protocols stain darker. Why do they stain darker? Well, they're tightly compacted, right? And as we mentioned, tightly compacted DNA is darker. It stains darker than the rest of the DNA. So you can actually sort of see that along the DNA, little bits and pieces that are slightly darker. So slightly darker blue areas. And again, that's actually tighter packed DNA. It's kind of a fun thing. So let's talk about our centromeres because they are important. Now, centromeres are constriction of chromosomes. It's bound by a complex of proteins, and they have a wide variety of functions, and they do help maintain sister chromatid cohesion during mitosis and meiosis as well. And they also serve as the attachment site of kinetochores, which, as we discussed, are just a type of microtubules, which is a really important part of our friend mitosis and meiosis. Now, centromeres have really interesting sequences. They have highly repetitive DNA sequences called sen sequences. And so where we have repeating sequences of TG dash, where dash can be any base, any of the four bases. And they have this repeating areas here. Now, the kind of interesting thing is that sen regions vary pretty heavily between organisms. So we can actually look at sen regions of our closest primates. So for instance, chimps or gorillas, they look very, very different than ours and vice versa. So kind of an interesting thing. Now, telomeres, they are at the tips of chromosomes. They're the caps of chromosomes. They're highly repetitive. And they're sort of the wild thing about this. And I don't quite think there's an answer as to why this is yet, but most vertebrates, so large animals, things with spines, of course, have the same repeat sequence of the telomeres called TTAGGG. Don't know why, kind of a wild thing, but it's non-usable pieces of DNA, so maybe it makes a little bit of sense that there is some conservation, but I don't know. Now, the final thing I'll make mention about telomeres before we move on is they do get shorter. Every time your DNA is replicated, as you probably recall, the telomeres get a little bit more shorter. So just a couple extra slides here for you about repetitive DNA sequences, kind of a cool thing, because as you'll sort of see here, when we look at exons, so these are the regions that actually code for genes that give rise to RNA or tRNA, you'll notice that we have are sort of lots of repeating areas, right? Tandem repeating areas, ALU elements, which are just repeating elements, introns, so non-coding regions. And so you notice that for your average eukaryotic organism, there's lots of sort of what we call junk DNA. It's kind of a weird thing. And this is actually in direct contrast to bacteria, which have lots, very, very few extra DNA. So we have a little bit of extra information here for you. Take a peek at it if you want to read a little bit more about repetitive DNA sequences. I think it is pretty cool. In particular, I think this little section here about microsatellites here that I make mention of. If you've never heard of those, you should take a peek at those. They're pretty cool little bits and pieces. So just as an optional, but again, if you're interested. Now, the final thing we'll make mention of about the packaging of DNA is actually where else it's found. Now, where else can this found is, well, actually in a couple of organelles besides the nucleus, which include the mitochondria and the chloroplasts. Now, mitochondria and chloroplasts, as you recall, likely arose from a symbiotic event of an algae and a heterotrophic bacteria. And so they have their own small circular piece of DNA. It's very similar to bacterial DNA in both structure and the proteins involved. And eukaryotes do have that. Now, the interesting thing about the human mitochondria, and it's been sequenced multiple times. And as you probably know, you get your mitochondria from your mom. So it does tell us a little bit about maternal inheritance. It's kind of a cool thing. But the human mitochondrial genome is only about 17,000 bases. It's 37 genes, and about 5% of all the RNA and protein needed are actually by the mitochondria. So it has its own genes, it has its own DNA, allowing it to sort of do its own functions. And sort of this is just a little bits and pieces of what that looks like, including some genes for ribosomal RNA, cytochromes, as you recall, cytochromes and all these complexes are involved with the electron transport chain. We also have ATP synthase and a couple other things. So really important piece of chromosome, but overall, um, just something to make note of. Now, chloroplasts are the same way, but they're just simply longer. Typically speaking, chloroplasts have about 100,000 plus DNA bases in their proteins. I'm sorry, in there for the genomes. So that's sort of the two other funky places where we see DNA. But let's talk a little bit more about our friend, the nucleus. Now, the nucleus is the site in which in our eukaryotic cell, the chromosomes are localized and where our chromosomes are replicated and the DNA can be transcribed, right? It's one of the sort of the characteristic things, right? So when we stain oops, sorry about that. When we stain a eukaryotic cell, we can see the nucleus. It sticks up pretty starkly, right? All over the cell, right? And what we sort of see is the overall structure is we have the nucleoplasm, so that inner area of the nucleus. So it's like the cytoplasm, but just inside the nucleus. Now you'll see the nucleus it's got a couple things. It's got a membrane. It's actually a double. It's a double membrane. So you see the inner nuclear membrane and the outer nuclear membrane. So it's a double membrane, and it has these pores. Now these pores are just proteins, as we mentioned in the membrane lecture, by which we can transport things through. And in particular, we're thinking about transporting messenger RNA through these. And you'll also notice the final thing is that actually the two things I'll make mention of is that they have the rough endoplasmic reticulum that borders the nucleus, as we saw previously. And then finally we have this nucleolus, which is just a funky little organelle that allows us to produce ribosomal RNA. That's sort of one of the things it does. So these are sort of just some information about sort of the things I make mention of about the structures of of our nucleus and things like that. But I do want to talk about what we call the nucleopore complex. Now, the NPC or the nucleopore complex is about 30 different proteins called nucleoporins, and it's actually it has an overall what we call striking symmetry. And you'll see what that looks like in just one sec, because it looks like it was almost man-made. That's how symmetrical it is. Now, there is what we call the central granule, and this is what we call the transporter, and it's actually involved with the movement of molecules across the nuclear membrane. So this is our nuclear pore. We have the inner outer membrane. We have, again, this really symmetrical protein, right? It's perfectly circular on both sides. There's the transporter protein. We have the anchor proteins, and we have this what we call basket of fibers. Essentially, just fibrous proteins. But again, really sort of fascinating structure, right? It looks very much like, almost like a pipe like a human would build, right? It's really sort of remarkable. And the final thing we mention of here is actually the size, right? We're talking about 120 nanometers, so it's not a huge piece of machinery here, but it is protein embedded in the membrane. And our friend, these nucleopore complexes allow us to transport a wide variety of things, right? Allowing us for pulling in proteins needed for transcription or proteins needed for our histones, right? Or allowing us to export the three distinct types of RNA for for translation or for some other process, right? So just some extra slides here about import and export of materials. It's a little bit sort of beyond sort of the scope of this class, but I figured I'd make mention of it because it's kind of a cool thing. So this brings us to part 2, which is by far the most exciting part of this class, and this is actually replication of DNA. Well, actually, the second most exciting thing. I think part 3 is by far the most exciting. And this is the replication of DNA because DNA needs to be replicated, right? We need to pass it on to daughter cells. We need to pass it on to the next generation. And so now that we know the structure of DNA, now that we know where the DNA is found, how it's coiled, and so on and so forth, let's talk about how we replicate this bad boy. Now, why do we care? Well, remember, if we're going to pass our DNA down to the next generation, it needs to be accurately replicated. Because if you're going to produce new liver cells, your liver cells need to know exactly what to do to be a liver cell, which means they need all the DNA inside that all other previous liver cells had. And that's where DNA replication comes in. Now, DNA replication is semi-conservative. This is proposed by Watson and Crick. So let's take a peek of it. We have a piece of DNA here. Remember, DNA is double-stranded. If it's replicated into 2 strands, the 2 new strands are comprised of half of old DNA and half of new DNA. This is what we call conservative replication. Now, if there's a couple other types that we really don't care about, but just do know that conservative replication is what we do. It's also what bacteria do as well for that. There. Now, if you want to know a little bit more about the discovery of DNA replication, it's really cool. Take a peek at it. But again, completely optional, but pretty cool. Now, DNA, as we mentioned, is bidirectional, meaning that it goes in essentially both ways at the same time, right? It doesn't just proceed in one way, it goes both ways. Meaning it goes in two directions. Now, this happens in what we call replication forks. So let's just take a peek at replication. We have a piece of DNA here. We have replication forks here. This would be an E. coli, just as a note. But you notice we have the origin replication. This is where replication starts, and we have this overall origin replication here, and we have replication forks where the DNA breaks down and we synthesize brand new strands of DNA here. You can see that here. You can see it here. You can also see it in this cartoon diagram here. So what we're going to focus on here is actually bacterial replication primarily because it's much easier to understand. But the reality is that there's a high degree of homology between bacterial and eukaryotic replication as well. Now, replication forks, in terms of our bacteria, do move away from the origin of replication. They unwind the DNA, and copying both strands of the Plasmids. This is what we call theta replication, and it's observed in replication of circular DNA molecules. Now, eukaryotic replication is a bit different, but pretty similar. Now, bacteria have circular chromosomes. Eukaryotes have linear chromosomes, right? We don't have circular chromosomes, right? Ours are linear. Now, in eukaryotes, our replication is actually initiated at multiple sites, and that's due to a couple things. Primarily because our genomes are so dang large, right? So if we're going to replicate 3 billion DNA bases, we got to start at a bunch of different spots if we're going to get through it, right? And so we have multiple areas and we create multiple replication units, what we call replicons. And at each site of replication, we have two replication forks synthesizing DNA in what we call a replication bubble. So we have a double-stranded piece of DNA here from our eukaryotes, multiple origin replication, forming replication bubbles. And eventually these replication bubbles fuse together, forming an overall larger one. And then we eventually have the overall replicons fusing together, allowing these two DNA strands to eventually split. So that's the sort of raw flow that we're going here. Right? Now, regardless of whether you're bacteria or eukaryote, we have multiple or single origin replications. And it's actually the way we initiate replication at these places is actually by groups of what we call initiator proteins. Now, the sequence that tells us, hey, where do we start replication is heavily enriched in adenine and thymine, and it's about 245 base pairs in length. You could think of it almost like a barcode. But the cool thing about this is it does vary pretty heavily. So there, among bacteria as well as eukaryotes as well, but it does vary pretty heavily. So let's take a peek at what we're going here, where we have the E. coli chromosome here. We have origin C. That's the area at which replication begins. Now you notice that we have a relatively small area here, not very, very large, at ORAC. So we have these what we call flanking sequences, and we have the consensus sequences, noticing that it's very heavily enriched in A's and T's and G's. Similar here as it is here, right? And so we have this overall similarity, again, recognizable areas that allows us to say, hey, this is the origin replication. Let's start synthesis here. Now, what initiates replication in our microbes? Well, there's a couple important enzymes we're going to need to remember here, and I don't expect you to remember all of them, but you certainly want to remember sort of the overall functionality here of replication in bacteria. Now, the three enzymes we're going to look at are DNA A, B, and C, so a little bit easy. Now, these three enzymes will actually bind and initiate replication at ORAC, so the origin replication. Now, DNA A binds to a part of the ORAC, which results in the overall unwinding of the DNA. Now, to stabilize single strands of DNA, now single strands of DNA, I don't think I mentioned this prior, but single strands of DNA are inherently more unstable than double-stranded DNA. So if you take double-stranded DNA and you make it into single-stranded DNA, you need to stabilize it somehow. And we can do this by what we call SSBs, or single-stranded binding proteins, which bind and stabilize our single-stranded DNA. So DNA A binds and starts getting DNA to unwind. It essentially relaxes DNA. Now we have DNA B, which is a DNA helicase, which actually begins to unwind or unzip the DNA. And then we'll get to our friend DNA C in just a moment. But the idea here is DNA A is oftentimes what we call a DNA gyrase. DNA B is what we call a helicase. So let's take a peek at here. We have OREC. We have the overall structure here. We have DNA proteins. It binds to that OREC. It causes it to essentially open up. I'm sorry, it causes it essentially decompress, allowing it to become more linear. Then we have single-stranded binding proteins that come in addition to DNA B, that allow us to essentially stabilize our DNA. And then we have DNA B come in. It starts to unzip our our double-stranded piece of DNA. And so that's the overall function here, right? DNA A binds. It causes the overall unwinding at that area. DNA C and B come in, initiating overall splitting of the helicase, leading to the initiation of replication. Now, what does this in eukaryotes? Well, it's not overall that different, but it is a bit more complex. Now, in eukaryotes, we have a multi-subunit protein complex, which we call the origin recognition complex, or ORC, or ORC, depending what you call. Now, this binds to the replication origin. And then next, we have what we call the mini chromosome maintenance. Try saying that 10 times fast. Or MC, which binds to the origin. So, now, once the MCM has bound, now, just to make mention before I move on, the MCM bounds, it has several DNA helicases, so it's doing the unwinding, so very much like DNA B does in bacteria. It also has a set of proteins called helicase loaders, which recruit the MMC proteins as well, or further, I should say. Now, at this point, when we have MMC and they're doing, and as well as helicases bound there, the DNA-bound proteins make up what we call the pre-replication complex, and then the DNA is what we call licensed or ready for replication. So once we get, and again, it doesn't really matter which organism we're talking about, once we get this bad boy open, we need a specific enzyme to catalyze the synthesis of a new strand of DNA, or strands, I should say. And this is an enzyme called DNA polymerase. It should sound familiar. Now, DNA polymerase has directionality to it. As you recall, DNA has a 5' and 3' end. And our friend DNA polymerase can only read DNA in a specific region. Now, DNA polymerase can only five prime to 3' direction. So let's take a peek at what we have here. We have a growing DNA strand here. We have the template DNA strand here, 3', 5'. It's adding in the 5' to the 3' direction. So it binds first to the 3' end, where there's a 3' hydroxyl group open. DNA polymerase, and you'll notice it consumes a phosphate group, so it requires energy, of course. And we have the adding to that 3' end. So we're adding from 5' to 3' on the new strand. That's the part people get tripped up the most, is we're not adding to the 3' end of the template strand. We're adding to the 3' end of the new strand. So we're going 5' to 3', but it appears like on the template strand, we're going 3' to 5'. So that's what you want to make mention of. DNA polymerase adds from 5 to 3 on the new strand, 3 to 5 on the old strand. Now, the thing I'll make mention of before we move on is that there's a phosphoester bond here, right? Phospho because phosphate, ester because there's an O in between two things, thus phosphoester bond. And that's what our friend DNA polymerase does. It doesn't just pull DNA bases in, it catalyzes the formation of a phosphoester bond. So just some handy proteins involved in DNA replication in bacteria and eukaryotes. You'll notice there's a multitude of them that are similar: primase, helicase, sliding clamps, so on and so forth. So just there for you to see. So, excuse me. So DNA is synthesized from 5' to 3' direction, but as you recall, there's 2 strands of DNA, right? And these strands of DNA are anti-parallel, right? So if I'm going 5' to 3' on one strand, well, the other strand's in the opposite orientation. And DNA polymerase only goes 5' to 3'. So this leads us to 2 distinct things that you probably recall: the leading strand and the lagging strand. The leading strand is just one continuous synthesis of DNA, whereas the lagging strand synthesizes things in small bits and pieces, or it's called, essentially what we call discontiguous DNA synthesis, and forming what we call small bits of DNA called Okazaki fragments. So let's look at this. Now, this is the same as it is in bacteria and as in eukaryotes. It doesn't really matter. Where we have overall direction of replication. So we're going this direction to this direction. We have the replication fork here. We also have the replication bubble here, but we'll stick talking about the fork because I think it's a little bit easier to sort of follow, at least in my opinion. Now, DNA polymerase, remember, it goes 5' to 3' on the new strand, which means it's going 3' to 5' on the old strand. Thus, if I need to go 5' to 3' on the new strand, it means I have to go 3' to 5' on the old strand, which means that on the top strand here, I'm working this way, whereas on the bottom strand, I'm working this way. So if replication is proceeding this way overall, it means I have one continuous strand, which is called the leading strand. And then if I'm going this way on the lagging strand, I have little bits and pieces called Okazaki fragments. So lagging strand, leading strand. Now we'll talk about how we stitch those strands back together in just a moment, but let's just make mention of a couple things. Now the first is proofreading. Proofreading is performed in the 3' to 5' direction by exonucleases. By the exonuclease activity, i.e. the removal of nucleases if something's wrong, of DNA polymerase. Now, overall, depending on the organism you're looking at, there's about an error rate of about 1 in 100,000 on DNA replication, 1 of 100,000, which is not bad, but not great, right? Think about the size of a human genome. It's 3 billion bases. If 1 of every 100,000 of those bases is wrong over 3 billion bases, that poses a big problem. And so because of that error rate, we can actually proofread our things. And our DNA polymerases can proofread things into a 3' to 5' directionality. Now, this is done by exonucleases, which again will help us, and you can sort of break down what they mean. Nuclease, as in an enzyme that acts on nucleotides. Exo, meaning they essentially cut them out or degrade them. So exonucleases degrade nucleic acids from the ends of molecules, whereas endonucleases essentially make internal cuts in nucleic acid molecules, but essentially both of them allow the degradation of our thing. So let's look at a hypothetical scenario here where we have a growing strand. So it's going 5' to 3'. So this has to be the leading strand, of course. DNA polymerase is adding DNA bases, but it makes an error, right? It has A, T, and then it tries to add a guanine, right? But it can't, right? There's a thymine here. That doesn't jive, right? Chargaff's rules tells us A with T and nothing else. So if that's the case, we have the exonucleases cut those bad boys out and degrade them, allowing us to fix the problem. And this is just an overall, essentially a schematic of what this enzyme actually looks like. It's got a thumb and a finger and a palm. So it's like a hand going around the DNA. I know that's kind of weird to think about, but that's the way it's going. And again, it cuts things out, degrades them, thus the problem is solved. Now, that's proofreading. Now, there is an interesting facet that we need to make mention of in the fact that to initiate DNA replication, we actually need a little bit and piece of RNA. And these RNAs are actually called RNA primers. So RNA primers bind to DNA, allowing DNA polymerase to bind to it. Without RNA primers, there is no binding of DNA polymerase, which is kind of an important feature. Now, if every time we start DNA replication, an RNA primer has bound, again, RNA primer is just a simple stretch of RNA. If you have a lagging strand, go back a couple slides. If you have a lagging strand, well, I have an RNA primer here, and an RNA primer here, and an RNA primer here. But on our leading strand, we only have one RNA primer. So if we have all these RNA primers, what do? Well, DNA polymerase does add nucleotides to the RNA primers as well as the DNA primers. However, cells contain an enzyme which we call primase, which can actually synthesize about 10 base pairs long. It varies, of course, because the primers vary between species, but they're short 10 base pair things of chains of RNA using DNA as a template. And this enzyme primase is used to initiate RNA strands without a pre-existing chain to add to. So our RNA primers, Baby crying. So our RNA primers are synthesized pretty straightforward. So we have primase. It binds to DNA, a single-stranded piece of DNA, and it's an enzyme. So it catalyzes the synthesis of a small bit of RNA primer. RNA is inherently unstable. RNA is inherently unstable, so we have to actually replace RNA. Now, who replaces it? Well, actually, DNA polymerase 1, right? So DNA polymerase simply replaces RNA with DNA, and we have DNA ligase actually seals the gap on our brand new strand. It's kind of a cool thing. So let's talk a little bit about how our DNA is unwound. Now, as we've mentioned before, DNA helicases unwind DNA. Um, and overall classes, there's lots and lots of different types of these, just as a note. And they do use ATP, or I should say the hydrolysis of ATP, to drive their capacity to unwind DNA. Now, in our friend Sorry about that. Had to change a stinky diaper. Fun stuff. So as I was mentioned, helicases act down here past the origin of replication. So let me go back a couple slides, back to where we were. Alrighty. Here we go. Here we go. And so in E. coli, so our favorite bacteria, we know that there are at least two distinct types of helicases. One binds to the lagging strand and one binds to the leading strand. So there are two things. Overall, when we combine two of these together, we call these primosome. I apologize if you can hear the baby in the background. He's still unhappy, even though I changed his diaper. That's just what babies do. Now, as we may mention, we have the single-stranded binding proteins, or the SSBs. Once the strand separating has begun, we have those bind in. Now, remember, once DNA is single-stranded, it's vulnerable, it's not stable. We have those proteins to come in and bind. And then finally, we have the Toposoma Races, which ease the super one side, but is on both sides, unwinding our DNA. And once our DNA is single-stranded, we have SSBs, or single-stranded binding proteins, coming through to stabilize things. Now, our riposome, so that overall replication complex, the winding and synthesis are coordinated on both strands via the overall together. Now, all these proteins involved with replication and winding and so on and so forth are simply what we call the riposome. So it's essentially a quaternary structure of a bunch of different proteins together. Now, the riposome itself is about the size of a ribosome, so it's a pretty sizable group of proteins, and it's powered by the ATP, of course, so the hydrolysis of ATP. Um, and because it needs energy. Now we can actually describe how it actually moves, and I love this little model. I don't know why, I just think it's so funny. Is actually the trombone model, where we have a sliding cramp protein that actually attaches to DNA polymerase, which is and it attaches the catalytic subunit. That's not super important, but that's where it attaches, and allows the polymerase to essentially process along the DNA without falling off. So this is this whole mass of protein. Right? We have DNA polymerase here, DNA polymerase here on the lagging strand, or the leading strand doesn't really matter. And you see we have our overall couple things. So we have the clamp loader, the sliding clamp, which is waiting to be loaded. We have what are called tau proteins that connect these two things. We have DNA helicase, we have RNA primase, and we have single-stranded binding proteins, and we have toposomerases here, or gyrases here, again, relaxing these things. Now, this overall mismatch of proteins is what we call the hollow enzyme, or simply called the riposome. Huge overall piece of protein, allowing us to synthesize DNA bidirectionally at the same time in a coordinated fashion. Now, after a stretch of DNA has been replicated, our nucleosomes, so remember those histones, have to reassemble them. Now we have a couple different proteins that are involved here in eukaryotes. First is NAP1, which is the nucleosome assembly protein 1, and chromatin assembly factor 1, or CAF1. Those are just two common and really important ones. But we have to reassemble back into a chromatin. Right? We have to go from a linear piece of DNA back into the supercoiling of DNA that we need, because remember, DNA is huge. Now, telomeres, remember, those are the end caps of our DNA. They're needed to solve the end of replication problem. Now, you'll recall that lagging strand lags behind, right? So we go lagging to leading, lagging to leading. So what we end up getting by the time we get to the end of our chromosomes is, as we go from the leading strand, it does one straight synthesis. However, by the time we get to the end of our chromosomes, where the telomeres are on both sides, of course, not just one, there's actually the last primers, when they're removed, they actually leave a little bit of a gap. And so what ends up happening is, once the last primers are filled, once the last primers are removed, we have this gap. And that gap is single-stranded DNA. So it actually will degrade via an exonuclease activity. And so after every round of replication of your DNA, the telomeres, or the end parts of the chromosomes, get shorter and shorter and shorter. Why? Well, because the lagging strands, again, the leading strand is all in one go, but the lagging strand has a little bit of a primer down here or here, depending which way you're going, and that part gets degraded and doesn't get fixed. So if you ever wonder why your telomeres get shorter, well, they get shorter because of the nature of the lagging strand replication. Now, human telomeres are sizable, about 100 to 1,500 copies, depending on the age of the person. They have typical repeats of TTA, GGG. These are at the end of the chromosomes. Now, these are non-coding regions. Again, they're just involved with solving the problem of the lagging strand, right? The extra piece of RNA primer. Now, there is an enzyme, it's called telomerase, which is one of the coolest enzymes out there. It actually catalyzes the addition of repeats to the end of chromosome ends. Now, for instance, and just as a sort of example, our friend tetrahyamina, which we got to play around with in lab, actually has a sort of interesting example here. Now, telomerase is a component of both protein and RNA, so it's not just protein. It's kind of a cool thing. And our friend tetrahyamina, the RNA component of the telomerase, is actually this sequence, which is actually complementary to the telomere repeat sequence of this. So as I made mention, complementary to this. And so what telomerase does in tetrahyamina is that it recognizes this sequence, I should just stay here, this sequence in the DNA, that open strand, and then synthesizes a brand new piece of DNA to match it. And so we can actually just sort of look at this. So we have telomeres here, we have the little bits and pieces that should fall off. We see your repeating units here. What telomerase does, it has a piece of RNA, acts as a little bit of template, matching what we see here in our DNA, allowing telomerase to synthesize a brand new piece of DNA, extending the DNA, essentially minimizing breaking. And telomeres are kind of cool because as the more and more replication you have, the more and more degraded they become. But the reality here is that as you do more and more replication, the more and more degraded your telomeres become. So the older you get, the shorter your telomeres get. And the sort of interesting thing about telomerase as an enzyme, it's actually completely inactive in most older cells. So younger cells in infants, it's pretty active. They're constantly dividing, thus they're constantly fixing their telomere problem with telomerase. But the older you get, the less it gets. And so you can actually see this. So we can look at fibroblasts. And so it's inside your body. Younger ones, nice and happy, looking good, versus older ones, looking kind of sad. So telomerase is pretty cool because telomerase and telomeres are involved with the idea of aging. Older cells, or what we would call senescent cells, they have no active telomerase. Their telomeres get shorter and shorter and shorter. Now you can imagine if telomeres are only so long, the more replication you do, the more you start to eat into the DNA that actually matters. And that's exactly true. And it's kind of an interesting thing is once cells typically lose their telomeres to actually undergo apoptosis, or programmed cell death. But cells have a limited lifespan, and that lifespan seems to be related to telomeres. And so there is a whole group of cells out there. I'm sorry, cells. There's a whole group of diseases out there. Um, I'll make mention of, including Werner syndrome, that actually are a result of too short telomeres. But we also know that cells that are immortal, so many of you probably have heard of HeLa cells from Henrietta Lacks, those are immortal cells because they never degrade their telomeres. Why? Because they've actively expressed telomerase. Now, it's kind of a cool thing about cancer cells in general is cancer cells are actually immortal. For those of you that don't know, HeLa cells are immortal cells. They're cancerous cells from a woman. And they're actually immortal because of telomerase. And cancer cells commonly are immortal because they have telomerase active. So kind of a cool thing that we'll see. So we'll take a break here. We'll pick up next class because I have to deal with the baby problem. We'll go over DNA damage and repair. In addition, we'll go over transcription as well. So we got a little bit ways to go, but we'll certainly go over more as we go. So hope you guys enjoyed something, and I'll see you around. Bye.